Transfer the MEA plate with the inner box into the clean bench. Remove the MEA plate from the inner bag and rinse each well surface with sterilized distilled water. Repeat the rinsing process 3 times. Rinse the upper lid 3 times as well. Pour 70% ethanol over the MEA plate and the upper lid for sanitization and leave for 15 minutes. Remove 70% ethanol from each well after 15 minutes. Rinse each well surface and the upper lid with SDW 3 times. Dry out the MEA plate and the top lid completely under UV radiation for 15 to 30 minutes. In subsequent processes, handle the MEA plate after drying by putting it in a sterilized container. To prevent evaporation of medium during incubation, place small dishes filled with SDW in the container. Dissolve 1 mg per milliliter human plasma fibronectin solution with DPBS to 50 micrograms per milliliter. Place the MEA plate on the MED Cell Droplet. Drop 4 microliters of 50 micrograms per milliliter fibronectin solution onto electrodes in each well. Make a 4 microliters droplet at the tip of the pipette tip and transfer it without touching the electrodes. To prevent evaporation of the fibronectin solution during incubation, wrap the square dish including the MEA plate with parafilm. Incubate the MEA plate in refrigerator, at about 4 degrees Celsius, overnight. Transfer the MEA plate coated with the fibronectin from refrigerator into CO2 incubator to incubate it for about 30 minutes. Remove the frozen cardiomyocytes vial from the liquid nitrogen container. And thaw the cardiomyocytes at 37 degrees Celsius for about 120 seconds. Gently transfer the thawed cell suspension, 350 microliters, into a 50 ml centrifuge tube. To collect remaining cardiomyocytes in the vial completely. Pour 650 microliters of warmed thawing medium into it. And then collect all the remaining together with the 650 microliters of medium. To avoid osmotic shock, Gently and slowly add the medium into the 50 ml centrifuge tube at a drop per 4 to 5 seconds. Add another 1 ml of thawing medium into the 50 ml centrifuge tube. Drip the first 0.2 ml over 30 to 60 seconds and the remaining 0.8 ml over 30 seconds. Count the number of viable cells. Gently mix the suspension and medium by inverting the tube 2 to 3 times. Centrifuge at 180 G for 5 minutes and then remove the supernatant. Add thawing medium to make the cell suspension 3 times 10 to the power of 4 cells, 4 microliters. Transfer the MEA plate from incubator into clean bench. Drop 4 microliters of cell suspension directly onto the fibronectin droplet on the electrodes. Replacing the pipette tip for each well makes it easier to form a droplet at the tip of it. But it is no problem to not replace it until it becomes harder to form. Incubate the MEA plate seeded with cardiomyocytes under 5% CO2 at 37 degrees Celsius for about 60 minutes. After incubation for about 60 minutes, carefully add 1 ml of maintenance medium into each well. To prevent cells from floating, carefully add 250 microliters at a time, repeat it 4 times. Incubate the MEA plate seeded with cardiomyocytes under 5% CO2 at 37 degrees Celsius. Replace half of the maintenance medium into new medium pre-warmed to 37 degrees Celsius on day 3. And completely on day 6, the day before data acquisition. Prior to placing the MEA plate, start 5% CO2 air aeration. And warm the stage to 37 degrees Celsius. Place the MEA plate with cultured cardiomyocytes on MED 64 Presto amplifier. Wipe any dirt or water droplets on the terminals of the MEA plate before covering. For drug treatment, remove the cover and administer the drug by pipetting. In this case, 50 microliters of medium is extracted first. Next, 50 microliters of the drug solution is added into the upper area of the well and agitated. And then 50 microliters is aspirated to add to the lower of the well to achieve the intended final concentration.